Thank you for that overtly generous introduction. I should mention that I'm also the Asa and Patricia Springer Professor of professorship that Tim endowed in the name of his beloved parents. And Tim, I should say, I'm so fortunate to be next door to you and learning from you, your insights in biology and your instinct in translating the science into real patient care. So I'd like to thank ASBMB leadership and the Vallee Foundation for giving me this incredible honor. I am especially humbled among the previous awardees that I listed here who have done amazing work in science. So my lab has been interested in immunity, as Tim has mentioned. And in recent years, we have been focusing on inflammasomes, which are, if I can call it, supramolecular complexes that activate caspase-1 or other so-called inflammatory caspases. And as you know, inflammation is a double-edged sword. It is essential for host defense against pathogens, but chronic inflammation, even when they are at a very low level, can contribute to diseases that are especially exacerbated by these modern societal factors. You know, we live so much longer, the environment is polluted, the diet, the obesity, and the stress. So part of our work that is very rewarding is that these pathways are highly relevant to prevent or treating these societal problems. And I call it cherry on top because by studying inflammasomes, we also reveal the molecular basis for the organizing principles, as what I call organizing principles in innate immunity through supramolecular assembly. This is something that, as Tim mentioned, that we discovered early on in my career. So these structures, sometimes they're amyloids, sometimes they're helical assemblies, sometimes they're simply multivalent assemblies. They're all polymers. That was something that I never heard of at the time. You know, why would these polymers be involved in signaling? It turned out these are everywhere in innate immunity, and now more and more in other fields as well. We had proposed that by assembly of everything into large molecular complexes, including the receptor, the ligand, the enzyme, the substrate, everything in one gigantic complex, you help to impart interesting properties to signaling, including threshold behavior because of the cooperativity, of course, signal amplification simply because they can polymerize, the temporal and spatial control of signal transduction because they give you a localization. And of course, on top of it, by bringing these signaling enzymes into proximity, you know, along these helical or other types of scaffold, you can enable very weak allosteric interactions to drive enzyme activation. So these common threads actually share mechanistic similarities to biomolecular condensate. I actually get this question a lot, that is, what is the difference between these more ordered assemblies that you observe versus the more liquid condensate? If you're interested in my opinion, please ask me after the talk. So, inflammasomes. So inflammasomes are these molecular complexes that I show here in the schematic, and many inflammasomes will form these gigantic speck in the cell, one per cell. And they assemble through activation by pathogenic or endogenous danger signals, which alter the conformation of the protein, and then it can recruit the adapter protein. Most cases is this ASC molecule, that's what we call it, ASC spec, and then ASC can recruit caspase-1. And within this assembly, caspase-1 gets activated, and therefore the pathway goes on. So here I show you some examples of what we've learned during the pursuit of these structures. It turns out these different inflammasomes use different molecular architecture, but they all undergo some kind of a conformational change. Oftentimes, it's a ADP or ATP-dependent conformational change, and their common downstream is always the formation of caspase-1 filament that drives the activation of caspase-1. And this also gives us the concept that in order to inhibit these pathways, which are highly pathogenic either through a genetic alteration in the pathway or through simply stress sensing, as you get old, you just get more stress accumulated through stress sensing. So there's, in order to keep them inactive, you need to inhibit the monomeric autoinhibited form. And this is what we are doing in the company that that I have co-founded, and they're undergoing clinical trials for a few of the compounds that they identified. So on top of that, so these so-called NR proteins in our pathway, most of them seems like do not have a lot of disordered region, but in one case, the NRP6 inflammasome is quite disordered. Similar domains, but quite disordered if you predict them. And it turned out they can undergo biomolecular condensate formation upon recognizing double-stranded RNA or this bacterial lipid, lipoteichoic acid. And then within the condensate, filament would form and execute the signaling process. So how about what the inflammasome do? So upon activation of caspases, they cleave important substrates. So interleukins are the bona fide substrates. They need to be converted to the mature form in order to become bioactive. Almost 10 years ago now, Feng Shao and Vishwar Dixit's lab showed that this protein called gasdermin D, it's simply a protein that that's expressed in gastrointestinal tract and dermis, the skin. Fairly unknown protein turned out to be the best substrate of these caspases. And we showed, I will summarize what we showed in terms of how gasdermin D helped secretion of the processed cytokines and also periapoptotic cell death. These are the two major consequences of inflammasome activation. So my former postdoc, Jianbing Ruan, and former student Xu Xia were the driving forces for elucidating the structure of just the N-terminal part, because the N-terminal part is supposed to be the pore-forming part of the gasdermin proteins. You can see they form these gigantic pores. I think this is the first time that anyone knows how a membrane can be damaged. So this is one way, is to form these large pores that are large enough to allow certain proteins to get out, including cytokines, even when the cells are not dead. And of course, they can contribute to the cell death process, and then more cellular components will get released from the cell. More recently, we showed that a particular residue in gasdermin D, cysteine 191, is palmotylated, is lipidated, which really explains how these gasdermin D can be recruited to the membrane. In fact, cleavage is not sufficient for gasdermin D pore formation. You really need this lipidation that drives the membrane association of gasdermin D and lead to the pore formation. And in this process, we surprisingly identified that even full-length, not processed, full-length gasdermin D can form pores, and they can be activated in response to certain cellular stress, such as reactive oxygen species-induced cell death. This is just to show you a piece of data that shows the N-terminal fragment is very active when it's palmotylated, and the full-length gasdermin D has certain activity and therefore can also drive the formation of pores. And this is a cryo-EM structure of the full-length pore with the green bits for the C-terminal part. And this is where cysteine 191 is palmitoylated right within the transmembrane region of the protein. So given that inflammasome activation and gasdermin deactivation is crucial for the induction of this highly immunogenic cell death, if you will. It's immunogenic because it releases all kinds of things to the outside. And we thought this may be utilized or exploited for anti-tumor immunity. So in this study, we identified small molecule agonist, which turned out to also act on cysteine 191. And the mechanism of which we deduced is that the small molecule mimic palmitylation and therefore activate the pore formation, and it's very potent in stimulate anti-tumor immunity. So we're pretty excited about this new mechanism, if you will, because the direct killing of cells or tumor cells by the agonist is very little, as shown. Oh, actually, I do have a mouse, as shown here. But this little bit of cell killing is able to bring the immune cells to the site of tumor and therefore control, leading to the control of the tumor. So this is, now we're coming to the part that I want to give you some detail. So I hope my time is okay. So gasdermin D formed these pores, but gasdermin D is not sufficient to induce cell death. It is sufficient to release the cytokines, but not for cell death. It turned out another protein called NINJ1, which was a cell adhesion protein. If you can't remember this, you just call it NINJA. It's a bona fide membrane protein with 2 predicted transmembrane helices, but has a extracellular N-terminal extension that's important for cell adhesion, but in the context of cell death, it's important for the damage of the membrane. And I will show you what we think, how this NINJ1 does membrane damage. So the most striking thing that came out of our in vitro reconstitution is that NINJ1 and NINJ2. NINJ2 is a related protein, but it cannot damage the membrane. So if we mix NINJ1 or NINJ2 with the membrane, you see NINJ1 there completely dissolve the lipids. The liposome is gone. Whereas NINJ2, when you mix NINJ2 with liposomes, it appears still form certain assemblies on the liposome, but it doesn't do anything to the membrane. So that was pretty striking. And when we try to solve the cryo-EM structure of NINJ1, as you can see, there are great variation in the size of the little ring-like structures, but we were able to take just small segments along the ring-like entities and determine their structures, because these smaller segments will have smaller variations. So it turned out, this is a segment that contains about 8 subunits. It turned out these assemblies all have the exact same kind of curvature. You can also see here in the 2D. All have the same kind of curvature. And when we modeled our protein inside these structures, it turned out that the hydrophobic bits of the protein faces the inside, whereas the hydrophilic part faces the outside. So this is completely different from how gasdermin pore looks like. It's the reverse of it. If anything, the membrane should be outside, and the outside would be hydrophilic. So how could that work? So here is the subunit structure. It has a pretty strong kink in the transmembrane domain. And then this bit here is amphipathic. And it should be extracellular. Oh, should be extracellular. This is a modeled structure because we don't know how it looks like before activation. But after activation, these amphipathic helix come down, and we think basically severs the membrane by forming these chain-linked ring-like structure through holding hands with the next subunit. So how do we know that this is true? So we reconstituted these macrophage that doesn't have NINJ1, but then we reconstitute them with the DOX-inducible NINJ1 GFP. During the inflammasome activation, you can see certain ring-like structures get formed, and then eventually they actually all get released. On the SDS page, the cell pellet shows that NINJ1 is there before inflammasome activation, but after inflammasome activation, it's gone. Where did it go? It went into the supernatant, essentially confirming or supporting what we observed through live cell imaging. So I think this is the killer experiment. So we took the supernatant, I meant my postdoc, took the supernatant and stained the supernatant with the Bodipi dye, which stains membrane. So NINJ1 already has GFP. What you can see is that these NINJ1 positive rings, and they all have lipid within, not outside, but within. So suggesting that these are lipid-filled disks, just like a nanodisk. So we call this cookie cutter. So just like a cookie cutter, it'll excise a piece of membrane out and release them to the cell supernatant, and just like a nanodisk that we use for membrane protein reconstitution in vitro. So my student did this ninja model of how ninjas would generate this and kill the enemy. So last year, another group from Sebastian Hiller's lab showed a different structure of NINJ1. The molecular assembly is similar, but in their case, their reconstituted structure is not in the ring form, but rather as double filaments, so two filaments together. And what's facing each filament is the hydrophobic side. So I think these structures are very straight, and this will be supposed to be the membrane-facing sides, but they couldn't know which side would face the membrane versus face the hydrophilic side. So they proposed a model that NINJ1 works like a gastrin D pore, but maybe a bit larger, or can damage the membrane through these filament structures. So I would say our model would suggest a somewhat different model, that is NINJ1 and gastrin D pores work completely differently, one through membrane damage by forming these pores on the membrane, and NINJ1, which is activated after gastrin D is activated. The signal of how it's activated after gastrin D activation is still unknown, but NINJ1 activation now induces membrane loss, not just the damage, but the loss of it. So then the cell is really in the state of lytic cell death with no return. So I'm really excited about the pathway and what it affords in terms of both the new biology as well as new ways to curtail inflammation. This is a fairly fast-moving field, and we hope to continue to contribute to it. And last but not least, I want to give a shout out to all my former trainees, the young people whose enthusiasm, whose innovativeness and dedication really made science so much fun. And I thank you for your attention.